All right, so welcome to episode 11 of the Breaking Bio podcast. I'm your host, Stephen Hamblin from the University of New South Wales in Sydney, Australia. Today is the 22nd of November here in the future. Yes, I know it's the 21st for you guys. And we have a full house today and a special guest, so I'm going to get right into it. And we'll start off with Lauren.
Hi, I'm Lauren Reid, a science communicator based in Glasgow.
Hi, I'm Michael Hawkes. I'm a PhD student at the University of Exeter.
Hi, I'm Morgan Jackson, PhD student at the University of Guelph in Canada.
Hi, I'm Tom Housley. I'm a PhD student at the University of Stirling in Scotland.
All right, and we have with us today our special guest, Mike Sivajothy from the University of Sheffield. And he's already laughing, so that's a good sign.
So, Mike, your research interests cover sexual conflict, and I was wondering if you could give our listeners a broad overview of what that is.
Yeah. Okay. Well, my interests are actually a little bit more constrained than that. I would say I'm really interested in how males and females interact, particularly during reproduction, and by that I mean actually during copulation rather than the whole stretch of reproduction. And my interests are kind of, I don't know, they've evolved over 20 years of doing research from trying to understand the structure and function of genitalia, through to really looking at how when insects mate and when genitalia interact, you often get wounding, and that stimulates the immune system of the insects. And so it's that interaction between the anatomical structure in males particularly, which interfere or damage with females, and how females respond to that, usually anatomically and or immunologically. So it falls under the broad umbrella of sexual conflict, which covers a vast span of different types of interactions over different types of evolutionary time frames. But my interests are really quite narrow in the context that we're talking about, and it's how males and females interact during copulation itself, and what the consequences of that are to both sexes, really. But inevitably it's the female, because that's where the damage is being caused.
So can you tell us a little bit about your main study systems that you work on this? Yeah,
so the system that we started, so about 15, 20 years ago, I noticed that there'd been actually an awful lot of work done in the 60s on bedbugs, particularly by a Frenchman called Carillon and an American called Usinger, and they'd done, as many entomologists in the 60s had done, they'd done incredibly thorough groundwork, and it was all, it looked really, really good, but it was in a context where it was in a 60s context. The way it had been explained was kind of completely out of left field, but the observations were actually incredibly tight and very, very thorough. And so I thought it'd be a great system. I was looking for something to do. I was getting bored with what I was doing, wanted to change in direction, and I thought bedbugs would be a really cool system to look at. Partly because they're easy to rear, you can keep them in the lab, and the labs, you can replicate pretty much what happens in the wild in the lab. So that struck me as a pretty good context to keep them in. And so we started working on bedbugs, and mainly because of their bizarre form of copulation, which is termed traumatic insemination, as you know, and the male actually wounds the female during mating. So rather than use the female's genital tract, he sticks his specially adapted Ediagus through the body wall and inseminates directly into a hemocoel. And all sumicids do this. It's one of the very strange features of the whole bedbug group. So that was the starting point, really trying to understand what was happening here, whether this was costly, what was going on, because there were all these stories flying around that males mated with males, and the sperm migrated through the recipient male and went to his testes, and then when he mated with a female, he would inseminate the female with some of his sperm and some of the parasitic male sperm And it just seemed it just seemed like it was going to be a laugh, really, and that's why we did it.
So is any of that true?
No, it's all bollocks. I mean, literally and metaphorically. No, it's not true. It's certainly not true in Simex, because Simex lectularis, the common bedbug, males very rarely mate with other males. So de facto, it can't be true. There are other groups. So apparently, in some xylocorids, males will what looks like intromission is happening, and so it may be possible that there's some transfer there. And in some Afrosimex groups, we know that males definitely mate with other males. So it might be true in those instances, but I suspect it isn't actually. One of the reasons I think it isn't is because we've done some experiments. They're not particularly top drawer experiments, but we've done some experiments where we've predicted what would happen if that was the case, in terms of the way the sperm would passage through the recipient male system. And you don't see the kinds of things happening that you would expect to happen if they were moving into the testes. So we can artificially inseminate, for example. Okay, we can take. There's a guy in my lab whose job it was to make glass penises shaped like bedbug penises, and we could use those glass penises to artificially inseminate females as well as males. So even though the males won't mate with males, you can set the you can't believe people get paid for this.
I'm sorry. I'm just imagining. I'm just imagining this little glass blowing setup. It's the tiniest little glass blower ever.
Well, it's actually, so what we do is we use an electrode pulling system. So it's a Narashigi electrode making system.
Oh, sure. Make it all scientific.
Yeah. Well, the beauty of it is you can control the length and width of the electrode that you're pulling, the glass electrode. So you basically set it up, turn the little knobs so that it makes a piece of glass that's the same dimensions as a penis. It's actually not that difficult.
I'm sorry, I really should let that one go. Go ahead, Tom.
How do you get the sperm to do the artificial insemination of bedbugs?
So what you do is you take you take the sperm from a donor male. You take it from his. The bedbugs are actually quite helpful with this whole process. So the males store sperm in a special kind of reservoir, and it's very easy to whip them open and get that sperm out. And then you just backfill the electrode or the glass penis with sperm and pressure it out, stab another male and pressure it out. It's pretty straightforward actually. It's a great good system for artificial insemination, right? Because it's easy to replicate a structure which injects a liquid into an open hemocoel. It's much easier to do something like that than it would be, say, to replicate mating in a dragonfly or in pretty much any other insect where the interaction between the genitalia is very complex.
So if you don't think these males are copulating with one another to kind of hijack each other's subsequent matings, what do you think they're doing?
I think what you've got here is a system of effectively scramble competition. So males, and that's what's driving this. So what happens is males can only mate with females when they've fed, when they've had a blood meal. And that's because the female goes from being very flat to being very fat. And when they're very fat, the structure, so the males don't just stab the female anywhere. There's a particular part of the underside of the female's abdomen where they can stab, where she's got a little groove in one of her sclerites. And she can defend that groove when she's flat. She can avoid being mated. But when she's bloated with blood, she can't defend that, and she's effectively a sitting target. So the males will only mate with recently fed females. And if the females, what the females are doing in this point in time, what most hematophagous females do actually, is they pump water out of the blood meal as quickly as they can, so they shrink down in size. And as soon as they get past a certain size, the males lose interest. So what's that got to do with males? Well, males have to blood feed as well. And when males blood feed, they go big and fat too. And so they look like females. They look like females because remember, this is all happening in the dark. And so probably what's happening is males, it pays males to jump on anything that's big and fat. And when it's and they basically, they're operating a kind of shoot first, ask questions later system. And the way they know whether it's a female in the dark is the only way they can do that is when they reach around with the tip of the abdomen and the penis feels for that little groove. And if the groove is there, it's a female. If the groove isn't there, it's a male. And in Cymex lectularius, at that point, the male will jump off a male. But it looks, to all intents and purposes, it looks like they're mating. So I think that's that's where the confusion came from. But it's not. The duration of mating when it's a female is obviously much longer than when it's males. So I think that's what's going on in Cymex. We're pretty sure that's what's going on in Cymex.
I'd like to take a break in here and welcome Bug, who showed up a little late, but we're all good.
Hey!
So yeah, Mike, this is Bug, the itinerant member of our podcast who sometimes shows up to talk about pubic lice. And
oh, great.
I have other topics.
I don't know. I've got some questions about pubic lice, but maybe we should.
Oh no! Let's start with that. What are your questions about pubic lice?
So, okay, well, I've got a story about pubic lice, which I'll tell you. It's a little anecdote. And it's got nothing to do with
Should we be afraid?
Well, I don't know. I mean, yeah, no, you shouldn't be afraid. It's not. I think it's perfect. It's safe. It's safe. I'd heard a story, and you can tell me whether it's true or not. I'd heard a story that pubic lice evolved with. That there's an element of local adaptation with pubic lice, and that pubic lice that you find on different races have evolved different pincer diameters because the pubic hair has different characteristics in different human races. And I thought, and I've been giving lectures and running practicals on adaptations in insects and tarsal claws, and I thought that would be a really cool way to get students interested in insects. So I rang up the local STD clinic and said, can I have some pubic lice? And they said, well,
boy, have we got good news for you.
And they said, well, actually, we haven't had any pubic lice for the last 5-10 years because there's a trend now that people remove their pubic hair and we just don't see them very often. But if we find any, we'll give you a ring. And this is in the hospital that's right next to to my building. So anyway, nothing happened for a year or two. I got a phone call. We've got a pubic lice. Would you like to come and get it? And we know, it came from a Chinese person, so maybe it'd be interesting to you. So I got really excited, put my coat on, ran into the section. This was at lunchtime. Ran into the STD clinic, and then suddenly realised, what the hell have you just done? You've just in the most busiest time when there are students walking everywhere, you've just gone into the sexually transmitted diseases clinic, and I then I thought somebody's bound to have seen me go in there. Luckily, nobody saw me. I got my louse, but of course, it wasn't any good because I only had the one louse, and I never got a chance to check that out. So, is that true? Have you heard that story before, or is it true?
Well, there's two stories.
Okay.
And are you guys getting reverb?
Should we maybe carry on, and then when Bug gets the mic working, we can cut back to the stories.
Yeah, I was just wondering about any female responses to traumatic insemination that you might have come across, if you pardon the pun. Looking at, can we edit that out, Stephen? That was a terrible choice of words.
No, actually we can't. I'm sorry.
I was just wondering about female responses in terms of their evolution to traumatic insemination.
So there's two responses. There's two levels. Okay, so there's a macroevolutionary response and kind of micro responses. So the macro level, if you look at female, so in Cymex lectularis, I said there's this little groove on the outside, and that's effectively that's the paragenitalia. So the female has a second set of genitalia which function as genitalia, and the so-called paragenitalia. In Cymex lectularius, the external manifestation of that is a little groove. The internal manifestation is a kind of complex and very interesting immunological organ. Now, if you look at the structure of those organs across different species, it's clearly evolved rapidly and divergently. So in most insects, you use the penis as a taxonomic tool because they evolve rapidly and divergently. In bedbugs, the penises all look the same, pretty much. It's the female genitalia that evolve rapidly and divergently, and that's probably what you see. Actually, that there's a real problem here because there isn't a good molecular phylogeny. The phylogeny is based on the female genitalia, paragenitalia. So there's a horrible circularity in trying to understand what was basal and what's more derived. But the primicymex, so-called primitive cymex, the male stabs the female anywhere. There's no particular structure. There's no internal structure, and then you go through a graded series, effectively, of complexity until you end up actually with species like Crassicymex and Strictucymex, African bat bugs, where the female has pretty much evolved a whole new genital tract, but that's linked up to this external system. So the male still stabs her, but he stabs her into a tube, and the sperm migrate through the tube, all the way through the tube, and then to the base of the genital tract, and then move up the genital tract. So you get this kind of bizarre reinvention of the genital tract, and so that, I suspect that's an evolutionary response to traumatic insemination, the evolution of this organ. If you take one insect, one species from that range of species, I think there's 68 species in the Cimicids, and our model obviously is Cymex lectularis, the common bedbug, and you look at how females cope with this system. They've got a number of ways of dealing with it, actually. One way is that because males only mate with fed females, there's a kind of association in the behavior, and females feed regularly. If you give them ad libitum access to a host, they'll feed every 7 days or so. If there's some discussions about whether it's 3 days or 7 days or whatever, but they'll feed cyclically, the very regular cycle, and of course it's females that are driving that behavior, right? So the female decides to leave her refuge and go and feed on the host. We thought it would be kind of cool to see whether that behavioral cycle was linked into switching on the immune cycle, because the female knows when she leaves the refuge to go and feed that she's going to get mated, because when she feeds she's fat, when she's fat she's attractive, when she's attractive she gets mated. And so those two events are quite predictable in time, and lo and behold, that's exactly what happens. So when females switch on their immune systems about a day before they go off to feed, so they're already up and running, anticipating the wounding effects and the introduction of pathogens or microorganisms into their body cavity when they go off to feed, so prior to mating. So that's a kind of neat physiological response to this. But there's another response which we don't fully understand. So males transmit in their seminal fluid something which enhances the females, or rather delays the onset of reproductive senescence in females. So there's in the same way that Drosophila males transmit seminal fluid compounds that enhance female egg-laying rate and simultaneously reduce female longevity, bedbug males transmit a compound which enhances female egg-laying rate but increases the onset of sexual sorry, the cats are fighting down here. Onset of the bedbug fluid. Okay, all right. Bedbug fluid increases egg-laying rate, but paradoxically also delays the onset of reproductive senescence, right? So that's another weird thing about bedbugs. They have a menopause, and effectively, it's the effect of seminal fluid will delay the onset of the menopause. So it has for some reason it enhances female fitness. So it looks like males are taking with one hand and giving with the other. They're stabbing females and causing all this wounding and immunological response. But with the seminal fluid, they're transferring something that enhances the female's fitness by increasing egg-laying rate and increasing the life expectancy over which the female will benefit from that egg-laying rate. So there's this weird thing going on, and we have no idea what that compound is. Yeah. So I kind of veered off the point there, but I've kind of answered your question.
Yeah, you have. Thank you.
Okay.
Sticking with the kind of evolutionary theme, for some of our listeners who may not know about traumatic insemination, can you tell us why it would have evolved that males spear females like this, rather than just copulating with them normally?
No idea. I mean, seriously, no idea. We've had, you know, there are times in your scientific career where you have absolutely brilliant ideas, and they're so brilliant and simple they have to be right. And you go and test them, and the first experiment you do, you think, yeah, I've really nailed this, and it just turns out to be bullshit. And I had one of those moments with exactly this question, right? So I was sitting there thinking, why the hell, what kind of thing would select for that kind of bizarre behavior? And it struck me that most female insects, bedbugs aside, store sperm in a cuticle-lined spermatheca, in a storage organ. So males have, and most female insects produce a spermatheca in the pre-adult phase as well. But there's no point in mating with pre-adult females, because when they close into adulthood, they're gonna shed the sperm that they've received in that mating. If you inseminate into the hemocoel, and the sperm can survive in the hemocoel, it effectively doubles the number of females you can get access to as an individual male. That was the idea, right? Because you can't shed your blood when you close. So we thought that would be that and you often see males attempting to mate and mating with pre-adult females as well as pre-adult males. So we thought this could be a reason why it evolved, that if you stab into the hemocoel that your sperm can persist. I mean, there's all sorts of other assumptions in there, but it kind of works, right? And we did this big experiment where we had lots of pre-adult females, and we put them in pots. We mated them, we put them in pots, and waited till they got to adulthood, and then saw if they laid any eggs. We did loads of experiments, loads of trials, where we had virgins, and just to make sure there was no parthenogenesis or anything like that. And in a couple of pots, we got baby offspring, and we all got very excited. But it turned out that someone had put an adult in the pot, and had screwed it up. But, you know, one of the technicians had put the wrong thing in the pot, and it didn't work. But that's the closest we've got to an explanation for this. It's very, very odd. It's restricted within insect or within organisms that have a separate male and female. It's entirely restricted to the simicids and a couple of groups that are closely related to the simicids. So there may be something ecological that's driving this, something about the way these animals live and make a living that predisposes them to this kind of thing. But it is very, very restricted in dioecious organisms. It's very common in hermaphrodites, traumatic insemination, but it's not at all common in organisms with separate sexes. So the answer to that question, how it evolved, is an open question. I don't think there's a good answer to that.
To follow up on that a bit, so we're very interested in this podcast about science communication and bringing science to everyone. And I wonder, not only for just pure science communication, but even for like grant writing and so on, how you explain why this is a cool idea to study traumatic insemination in particular. I mean, I know it's cool because, you know, as Tom would put it, stabby cock daggers, right? But,
okay, so the way this is actually the way this is cool is, to my mind, is actually less to do with the sexual conflict side of the story, but it's more to do with the cellular immunity side of the story. And the explanation on that side goes something like this: that, you know, 50, 60 years ago, we discovered CD8 and CD4, these cell surface markers that allow us to identify T cells and B cells. And that completely revolutionized our understanding of vertebrate immunity. Okay, before then, those were identical cells. There was no difference. You looked at the vertebrate white blood cell population, and there was this cluster of cells that kind of did stuff, but nobody was really sure what was going on. The second those markers were discovered, you open up an understanding of the secretion of immunoglobulins, T killer cells, all of that cool stuff that comes with the vertebrate immune system came from the identification of 2 cell surface markers. That was 50, 60 years ago. Insect cellular immunity at the moment is still based on this kind of clunky old phenotypic classification. You go and read even papers that were published this year on insect cellular immunity, it's about this cell looks like that, this cell looks like this, therefore they have a different function. I mean, I'm serious. That's the level at which we understand insect cellular immunity. Well, so what? Why is that important? It's important because 600 million people worldwide are affected by diseases that are vectored by insects. And those diseases are evading the insect cellular immune system. And we do not know how it works. I mean, that strikes me as utterly bizarre. And what's that got to do with bedbugs? Well, if you try and understand, one of the reasons it's taken such a long time, or taking such a long time to understand insect cellular immunity, is because they slosh around in this system. There aren't any organs. It's very difficult to get a handle on a system that you can actually look at and see how the cells function. But bedbugs provide that opportunity, because underneath this little groove on the hemophilic side of that groove, the female has a bag of immune cells. It's the only insect to have an identifiable immune organ, A, and B, a structure that's big enough that you can actually see with the naked eye, dissect and put in a petri dish and see how the cells behave within it. So it actually provides us with a really cool tool to try and understand how cells are coordinated into an immune response. And even better, we actually have the context of that response, right? That's the other problem when you look at immune systems, is how do you know that what you're looking at is a kind of natural response? Well, we know what it is in the bedbug, because all you have to do is stab the thing with a dirty penis, and that's what it evolved. That's what it evolved to cope with. So you can replicate this in the lab very easily and see what. Doesn't always laugh?
Because we're really mature.
Can I just say that, you know, if this whole science thing doesn't work out for you, you have a great entry to porn. This is good stuff.
Well, you've sold the subject to me.
Well, yeah, thank you. At least someone's taking it seriously. the point.
We take it very seriously here.
The point. No, I know you do. I know. The point is this. You know, I think you have to. The way you sell science to grant awarding bodies, and then this is a sad statement actually, is you have to be creative in the way you communicate it. You know, the thing that is actually quite interesting here is of course the stabby penis stuff, but nobody's going to pay for that. No one's going to give you money for that. You have to, these days, it has to be contextualized in a setting that has a value, an immediate, the so-called impact to the people who are paying for your science. And I think, you know, actually in some respects I feel obliged to justify it in those terms.
I think we all do.
Yeah, but a lot of people in my generation don't. They're very angry about having to compromise their their intellectual freedom to do whatever they want. And well, there's an element of truth to that, but it's how you repackage it. It's how you think about what you're doing and put it in a bigger context. And that is often very important, I think.
I worry about that myself. I mean, I'm a theoretician who spent my PhD studying social foraging models. Like, you know, trying to package that up into a ball that will please people in the public or granting bodies as to the massive importance of why this is going to solve cancer. You know, it is difficult, but we do have to do it. It is a part of what we do.
And it's a challenge, you know, and it's actually fun. You know, the money I get is always on the back of the cellular immunity, looking at immune function. But of course, once I've got that money, it allows me to do some of the cool stuff on the back of that.
Hilariously, I was watching a video that I found earlier when I was doing some background reading on the internet, and it was a guy from Genesis Week, who was, which is a creationist view of current scientific literature, and he was talking about some of a study that you'd done, and he claimed in it that you only get grant money if you put the word evolutionary in your grant title, and I was just thinking, that guy does not know how science works.
No, I think, actually, Tom, I think there were better clues than that. I think there were bigger clues than that in that video. That was hilarious, though. Yeah. So that was in relation to a study that a master's student of mine did, a very talented master's student, and it was a very simple experiment. She just wants to know whether fine hairs actually help to detect foraging hematophages. And it was, you know, it's as simple as actually having this. It's a pairwise design. You take the same person, shave their arm, and you put the bugs either on the shaved arm or the hairy arm, and ask them to tell you when they detect movement. And not surprisingly, the hairier you are, the easier it is to detect bed bugs. So it was a very simple. A very, very simple master's project. It actually turned out to be quite complicated in the design and the execution, but it was a laugh. It was a great little project. It worked, and it got a lot of press, and it also then, of course, got a lot of people's backs up, particularly the creationists who didn't like this notion that the human condition. You know, there were a lot of newspaper articles about, you know, we came from apes and apes are hairy, and that's kind of what got their goat, actually. That there was this link with the human historical past, a link we weren't interested, by the way, in, but that got kind of picked out in the popular press. Yeah.
I was just going to comment one last thing. I think it's really cool and more than a little ironic that a lot of your grants are based on using bed bugs to find answers to these immune questions for insects that are going to potentially save, you know, millions of lives, potentially, if they pay out. But you're using an organism that pretty much everybody on the planet cannot stand, and it lives in fear of whenever they go, you know, more than 20 feet away from their own house. Yeah. You know, in the way of the bed bug. So I think it's going to throw a lot of people for a loop when they hear that, oh, I'm using bed bugs to save you from, say, malaria. It could be. You
know, the other irony in that, it is weird, and the other irony is that you can't actually, if you say that I want, you know, that bed bugs are a major pest, and that that funding bodies should be funding research into them because of that problem, you know, particularly in the States. Actually, they're causing huge social problems in vast swathes of the eastern and western seaboard. The grant awarding bodies aren't interested. It's not on the political radar at all, because they don't transmit a disease. They're clean, right? And so there are lots of little ironies wrapped up in this, that you can't get money to work on them because they're a pest, because they don't transmit a disease, which is utterly bizarre. I mean, I just find that very odd. Pubic lice.
Damn it! Oh, Bug, you're killing us here. Come on.
Oh well, at least we now have an esteemed professor shouting pubic lice down the camera at us.
an ex-esteemed professor, yeah.
Wow.
So one thing that I read on your website, I think, was something called prophylactic immunity, and I was unclear about what that meant. Can you go into some detail on that?
Yeah, sure. So one of the other cool things about insects is that they have mechanistically, they have a very simple immune system. What we know about it, it's pretty straightforward. There's nothing sophisticated like the vertebrate system. There's no fancy molecules that can detect the shape of epitopes and educate other cells to only really blah blah blah. The whole immunoglobulin story. It all looks very simple. There's a whole bunch of soluble proteins that basically kill stuff, and there's a whole load of cells that basically swallow stuff up, and there doesn't seem to be very much sophistication. But what's actually happening is so a number of very clever people there's a guy called Paul Schmidt-Hempel in Switzerland and a couple of other groups that realized actually that the way you should look at immune systems, or an interesting way to look at immune systems, isn't to try and dissect them mechanistically, which is of course what everyone in the vertebrate field does, but to look at how they're managed, because you can do cool stuff with simple effectors if you manage them carefully. So there was this kind of trend about 10 years ago to start looking at what was called, and is still called, ecological immunity, and most of what ecological immunity does is try to contextualize immune function in a life history context and in a management context, how you manage the whole immune system. So rather than just taking cell X and seeing how it responds to pathogen Y, what you do is you take the whole immune system and see how it's managed over the course of a natural infection, right? So that's the preamble. So what does that got to do with prophylactic immunity? Well, people noticed, a guy called Mark Reeson and Ken Wilson worked on armyworms, and they noticed that when you had outbreaks, and this is true actually in a lot of phase-shifting insects, so solitary and gregarious phase locusts show this similar pattern as well, that in the gregarious phase they tend to be darkly coloured, and people often, and in the solitary phase they tend to be quite camouflaged and green or lightly coloured. And the explanation there was that on the solitary phase you should be camouflaged, and when you're gregarious you should be aposematic. And that's probably true. But the darkness, the dark cuticle, also is linked with switching on a whole load of immune effector systems, internal systems. So the same thing that makes cuticle dark is one of the key humoral immune systems, a phenyloxidase cascade, an enzyme cascade that produces melanin. So there's this link between the way that animals that are reared or that grow up at high density express cuticular and immune phenotypes in the adult phase, and the same species that's reared at low density expresses those phenotypes. So what Reeson and Wilson did was they took armyworms and reared them at different densities under sterile conditions. So the obvious explanation here is that at high density they're getting infected early on, and what you're seeing is a consequence of infection. But what Wilson and Reeson did, and what a number of other people, including us, did with different insects, is exactly this experiment: rear them at high density, sterile as larvae, and see what happens in the adult phase. And what happens is high density larvae produce adults that have invested more in their immune systems than low density larvae. And the explanation is simply that you're more likely, so this is prophylactic immunity, because you're more likely to be infected if you grow up with a lot of conspecifics next to you. It's as simple as that. That's the thinking. But what it actually provides is a really cool way of manipulating investment in immunity without having to challenge the insects immunologically, because that's always a confounding factor in experiments. So it provides a really cool tool to look at what the effects of immune investment are, and dissecting out the usual confounding variable there, which is that one lot has had a pathogen challenge and the other lot hasn't. So that's what prophylactic immunity is in a nutshell. Does that make any sense?
Yes, it does, and it also possibly explains why this huge experiment I set up last year, everything died as they were all isolated from birth. So I finally have an answer to why my PhD went down the drain.
I'm sorry to hear that.
So if we started recording the podcast a year ago, you'd have been a lot better off?
Yeah, that would have been great.
So what happened? All your low-density animals all died of infection?
Yeah. Well, everything was low density because I was isolating every single larva within 24 hours of hatching from the egg and rearing them up individually. And I'd done it the year before with, but mostly with inbred lines, and this time it was a huge breeding experiment that I'd done. And I was speaking to the guy who's the kind of world expert on my study species, which is the decorated cricket Gorillodis sigillatus. The expert is Scott Sakaluck, and he was saying that they actually have a lot less, much fewer problems with disease with the inbred lines because they're just weird and like an awesome line that's been going for 45 generations, whereas the
and it's probably really in a
population,
bucket, right? It's probably hundreds of them in a bucket.
Yeah. So that's good.
Breaking Bio, connecting people and solving problems.
Yeah.
Too late.
All right. Now, speaking of solving problems, I know everybody here has been waiting for the answer to this pubic glaze question. So let's get this. Come on, Bug. You have sound. Do it.
I do. And everybody hears me, right?
Yes.
Yeah.
Thank God. Okay. So what I was going to say was there's kind of two questions in the question that you posed about pubic lice. The first one is there's been a lot of talk about whether the sort of fashion for taking all your hair down there off has caused a decline in pubic lice populations. And there's been a couple publications, but they're really not very strong. When you look at their evidence, it's pretty much people measuring in a public health clinic, actually, in the UK. This is how many pubic lice we've had over the last 20 years, and it's pretty much a straight line down. And then they're also saying, and we know from the media that people are shaving their curlies off. But they can't connect the two other than just, we hear there are a lot of people that are yanking their hair off. So I think there's, and honestly, I think it's actually not entirely a function of people deciding to sort of go with whole-scale deforestation below the waist. I think it's really a two-pronged, if you will, factor. That one is, people are intensely grooming their shrubbery, and so in order to do that, in particular, if you're going to have, even if what you're going with is sort of the weird vertical landing strip, which I've never really understood, and even if you're going to do that, that means basically there's a whole industry now, and this industry is involved in having some stranger stick their head in your crotch and pull all your hair out. If at the time you also happen to have small animals running about down there, that's going to get noticed. And so I think the fact, it's not so much that we're taking the hair out, it's that we're paying intense attention to our genitals right now. Me being the rest of the population, I am not. Just so everybody knows, I grew up in the 70s, not going to happen. And so to follow up on that overly sharing comment.
Yeah, that was a bit too much information, actually.
But that's okay, he can edit it out.
Not a chance.
Ken and Will are two different things here.
Well, we got another one for the blooper reel then. So there's that, but also I think that, and since I mentioned the 70s, that's when pubic lice really hit their peak, and that's when a whole lot of people were having a whole lot of sex. Actually, it was a pretty fabulous decade. You guys really missed out. And except for the clothes.
I didn't.
Right. You know what I mean, right? It was awesome, wasn't it? Except for the double nip.
So the one of the interesting things, I guess, correlate. So the problem is we're dealing with correlations, right?
Right. Exactly.
And what I suspect one of the biggest drivers was actually the pre and post AIDS events.
Exactly. That's where I was going.
Okay.
Yeah.
All right.
That's where I was
going. Before you threw out any more euphemisms for the removal of pubic hair, I thought I'd stop you in your tracks there.
But I was
stop you on the landing
fun.
strip,
I've got
as it were.
6 or 7 more, really. But no, I mean, I think that's it. People don't have the kind of sex that they used to have. Okay, let's try that again.
Yeah, good.
As well. Somebody help me out here. You know what I'm trying to say.
No.
You got into the message yourself. You can
People are more
yourself
aware of their sexual health now, perhaps.
Well, there's not as much promiscuity.
That's a good way of putting it.
Yeah, it even sounds vaguely sciencey. But no, I think that more than anything else is a big factor. That people just aren't. You have to have transmission, and I don't think people are having as much sort of random hookup sex or unprotected sex than they used to.
Yeah. Okay. So that's answered that question, but that's not the question I asked. The question I asked
the different races.
Yeah.
I have never read that anywhere.
Okay.
It seems. I mean, I honestly am not so sure that I don't know. I mean, I would really like to know now if there is in fact a paper about the differences geographically in how people's pubic hair diameters, because that seems like where you have to start.
That's definitely true. So so hair. Okay. So there's definitely a racial difference between hair types and hair structure globally. So that's I don't know who measured.
Well, I wasn't really
the pubic side of
interested
that,
globally. I was meaning genitally.
Racially.
But sure.
Yeah.
Yes. Okay.
Yeah. But that geographic. I mean, it's an easy thing to test, right? I mean, all you have to do is collect pubic hair and pubic lice from different geographical regions.
I will totally do a postdoc with you on that if you can find some funding.
It's the funding, but we got to. You see, we can't. We have to package this in a different way. We can't. I can see this
public
causing all
health.
sorts of.
Public health. We could fold the shaving thing in there somewhere.
Best proposal ever.
I'm just waiting until they do the human care protocols on this one, the ethics board.
Oh yeah,
we
the
need some
IRB
undergrads.
for this is going to be awesome.
Yeah.
We need to shave some undergrads, and then we need to give them pubic hairs. This is totally cool. We'll give them $5. It's totally good.
So we're gonna have our face in people's junk. Let's do this.
No, let's not really. Let's not. That part needs to be undergrads. That's good.
All true.
Wow.
Bug, that was beautiful.
Yeah.
That was poetry. The last 10 minutes of this.
The sheer number of forestry metaphors you used to get that.
That was awesome. Yeah.
It was a thing of beauty.
Hey.
So, Lauren, anything to add from the call center experiences on pubic lice?
Yeah, for those who don't know, I used to work for a sexual health helpline. I've had many a story about a pubic lice and shaving, and as far as I know, they're still around because people are still phoning me to help with it.
Well, actually,
I've been call center for a long time, though, right?
I just do it voluntarily now. I just love it. Public service.
I should say that when I was looking this up, as one does, it turns out that actually probably only about 30% of the U.S. population, and I think something like 25% of Australians actually shave the no-no-fro, if you will.
Is somebody writing this down? We should have a lexicon of definitions here.
This is better than Urban Dictionary.
I'm in shock. Yeah.
So the vast majority of the population still is has
natural
intact shrubbery. Yes.
An overgrown lawn.
Climax vegetation.
Yes. The carpet and the curtains still match.
There are snow.
Haven't got for hardwood floors. How's that? Oh yeah.
Yeah, I think you're right, Bug. I think people are just more vigilant now. No one wants crabs. So
Mike, I am so sorry if Tom lured you on with the promise of real science.
Yeah.
There was totally real science until I got here. And I can go away again too if you want to get back to that. But I will say I am speaking. This is real data from published papers. It's just presented, in my own special idiom.
You're just bearing the facts, is what you're doing. Is all you're doing.
Yes, that's right. The naked truth. To get us back to bedbugs, actually, I have a bat question, because I know when I lived in the Southwest, a major issue was bat bedbugs, where what would happen is an exterminator would come in, they would remove a bunch of bats, and then about 2 weeks later, suddenly bedbugs everywhere, except that they were bat bugs.
Yeah, Pipistrelli, yeah.
And I just the question was, how common is that they'll go off and seek another host?
So it's interesting. So there's two species which do that a lot. One is Simix pipistrelli, which is the one you're talking about, and there's one that lives on martins as well, in martin nests, house martin nests, and the house martin ones invade home invaders when the martins fly off for the winter, and I think that's kind of probably the most likely, or one of the most plausible explanations for where Simix lectularius came from, actually, in our historical past. That we lived in caves or in houses that had bird nests, and they evolved from that line. It's all Simix anyway. It is common. It's very common, and it depends obviously on the density of bats, because that ultimately defines the size of the population of the bugs. The bigger the population of the bugs, the more likely they are to invade the house and feed. It's as simple as that.
And
I don't. What can I say?
Great,
Well, that's a good high note to end on. So, we're going to wrap up here.
Okay.
So I want to thank Mike for joining us today, and
I'd like to thank you for having me.
It was a pleasure indeed, and I am so sorry for all the forestry metaphors.
Yeah, that was interesting. Yeah, but yeah, thank you. They were great. I guess you're gonna post this online at some point, so I can get my notebook and write them all down.
Yeah, you should see it in a few days.
Brilliant.
But, all right, so thanks everyone for joining us, and if you want to wave goodbye, thanks for episode 11.